So, my dear viewers, you are all warmly welcome to our Vidhyalakshmi YouTube channel. I am Mrs. Lata from Botany department. Today we are going to see the very interesting topic, that is a comparison of microsporogenesis with megasporogenesis. So, what is the meaning of microspore? So, the microspore is considered as a male part of the flower. Okay. The megaspore is considered as a female part of the flower. So, first we know the floral parts. So, we know the floral parts, that is the essential and non-essential part. The essential part are, that is the androecium and gynoecium. Okay. The androecium consists of a number of stamens. Okay. Each stamen consists of a anther and the filament. Okay. Inside the anther, so it consists of a microspore. Okay. So, the microspores are developed into the pollen grains. Okay. So, today we are going to see how the microspore is developing into the pollen grains. So, that process is known as a microsporogenesis. Okay. Similarly, the megaspore, the formations of the megaspore, the formations of megaspore, so from the, so that is the female gametophyte. Yeah, the megaspore is which gives rise to the female gametophyte. Okay. So, what are the difference between the microsporogenesis and megasporogenesis? Microsporogenesis means that the stages involved in the formations of the haploid microspores from the diploid microspore mother cells. So, what is the meaning of the haploid? Haploid means that is a N, we consider as a N. Okay. That is a half of the chromosome. The number of chromosome is half from the diploid. Okay. The diploid microspore mother cells through the meiosis. Meiosis, it means that is a reduction divisions. Okay. So, that process is known as a microsporogenesis. So, what is the megasporogenesis? The process of development of a megaspore from a megaspore mother cell is called a megasporogenesis. Okay. So, how the microspore is developed inside the anther, that is we are going to see now. So, that is a development of the anther. Okay. So, we can see the different stages in the development of anther. The first one, that is the anther primordium. So, in the anther primordium, we can see that there is a early development of the anther. The early development of the anther, we can see a mass of cells. Okay. That is a homogeneous mass of cells. The homogeneous mass of cells are covered by a single layer. Okay. The single layer is known as, that is the so, the mass of cells are considered as a homogeneous cells. The homogeneous cells are, that is covered by a single layer. The single layer is known as the epidermis. Okay, that layer are known as the epidermis. This layer is known as epidermis. Okay. So, it means this is considered as a homogeneous mass of cells. Okay. The homogeneous mass of cells are known as a hypodermal. Okay. In the hypodermal cells, during the development, we can see that's a four-lobed structure. We can assume the four-lobed structure. It consists of your hypodermal cells. Each hypodermal cell should be act as your archisporium. That is act as your archisporium. Okay. The archisporial cells are undergoing the divisions. So, what type of division is occur in the archisporial cells? It consists of, that is a periclinal divisions. Periclinal divisions. As a result of periclinal division, which gives rise to the two layers. Okay. So, one is looking towards, one is formed towards the epidermis and another one is formed towards the sporogenous cells. Okay. Which gives rise to the primary parietal layer, that is towards the outside. Okay. And another one is a primary sporogenous cells, that is towards the inner layer. Okay. So, the primary parietal layer is again undergoing the divisions. Okay. So, what type of division is occur in the primary parietal layer? The primary parietal layer is undergoing the anticlinal and periclinal divisions. So, what is the meaning of anticlinal and periclinal divisions? Okay. So, the division is, that is your parallel to the neighbor cells, that is known as a periclinal divisions. The division is, there is a perpendicular to the parallel cells, that perpendicular to the adjacent cells. So, that is known as a anticlinal divisions. Okay. So, the periclinal anticlinal division takes place in the primary parietal layer, which gives rise to the four layers of the anther. So, that is considered as a anther wall. Okay. So, how it is formed? This is formed from the periphery to the center. Okay. So, what is the outermost layer? Already we know that is epidermis. Epidermis is the outermost layer, so that is already formed. Okay, so next layer is the endothecium. Okay, that's from the periphery. Next layer is the middle layer. Another one is the tapetum. The three layers are formed from the parietal layer. Okay, when the primary parietal layer is undergoing the division, there is an anticlinal and periclinal division which gives rise to the three layers. That is the anther wall. Okay, so except epidermis, the epidermis is the origin of archisporium. Okay, and remaining three are that is not originated from the archisporium cells. Okay, so what about in the primary sporogenous cells? The primary sporogenous cells, so that is undergoing the divisions. So what type of division is occur in the primary sporogenous cells? The primary sporogenous cells are undergoing the mitotic divisions. Okay, as a result of mitotic division, which gives rise to the primary sporogenous tissues are formed. From the primary sporogenous tissues, that is your microspore mother cells are formed. Okay, the first one, so that is a sporogenous tissue. The sporogenous tissue which gives rise to the mitotic division to form primary sporogenous tissues are formed. Okay, primary sporogenous tissues is undergoing the meiotic divisions. Okay, so that is formed into the primary sporogenous tissues are undergoing the division, that is also in the mitotic division to form a, so that is a primary microspore mother cells, MMC should be formed. Okay, enna form agum, MMC form agum illaya. So as a, from the MMC, that is a haploid microspores are formed. Haploid microspores are formed. Okay, so what type of division is occur between that? What type of division is occur on the microspore mother cell? So that division is a meiotic divisions. Otherwise, that is known as a reduction divisions, meiosis. Okay, the division name is meiosis. As a result of meiosis, 4 haploid microspores are formed. Okay, so this is the way the microspores are developing inside the anther. Okay, so that is known as a development of anther. Okay, development of anther. Here we are going to see the transverse sections of the anther. So what are the portions in the anther? It consists of your connective tissues, epidermis, endothecium, middle layer, tapetum, and next one is a stomium. Okay, the stomium is very important layer. Okay, so where we have to see the stomium. Stomium is present between the two lobes. Okay, actually this is here two lobes. 2 locules are there. It consists of 4 locule. Okay, so between the 2 locule we can see the very thin layer. The thin layer is considered as a stomium. Okay, so after the maturity, the pollen grains are dehiscence through this via. The stomium is it is giving the way for, that is the releasing of the pollen grains. So the development of anther, it is a very short way to recall. That is the anther homogeneous mass of cells. Actually, it consists of the homogeneous mass of the cells. So that is known as a hypodermal cells, isn't it? The hypodermal cells are act as a archisporium. The archisporium is undergoing the division. So what type of division is occur in the archisporium? So that is your periclinal division. So what is the meaning of the periclinal division? The division is occur, that is your parallel to the neighbor cells, okay, adjacent cells. And next, as a result of periclinal divisions, primary parietal cells and primary sporogenous cells are formed. Okay, from the primary parietal cells, the 3 layers are formed. That is the endothecium. Okay, that is your middle layer and tapetum. From the primary sporogenous cells, actually microspore mother cells are formed. Okay, as your division of, that is a meiotic division. Okay. So this is the formations of microsporogenesis. Okay. So what about in the primary sporogenous cells? That is undergoing the mitotic divisions. So in this chapter we must keep it. So all the type of divisions where which takes place, that is very important part. Mitotic divisions, as a result of mitotic division, which gives rise to the sporogenous tissue. Okay, sporogenous tissue. From the sporogenous tissue, microspore mother cells are formed. Okay, so microspore mother cell is undergoing the meiotic division to form a, so that is a microspore. Okay, that is a 4 haploid microspores are formed. You can see very clearly here. So in this picture, you could easily understand. So you can see this is a meiosis. Okay, the meiosis takes place in the MMC. Okay, that's a microspore mother cells. Okay, as a result of meiosis, that is your tetrad of pollens are formed. Okay, the tetrad of pollen grains are, that is the pollens, actually the microspores are formed inside the anther. Okay, so now each and every microspores are segregated from one another, that is a release from one another. Okay, and after the maturity it will be released. So the microspores are well developed into the pollen grains. Okay, so where the pollen grains are formed from the microspore. Okay, the microspores are early development of the pollen grains. Okay, see in some cases the microspores after the maturity, that is present inside the anther. Okay, so they have to held it together. So all the microspores are held together to forming a single bag-like structure. So that is known as a pollinium. Okay, so this type of pollinium we can see, this type of microspore we can see in the some of the species. So example for the calotropes. In the calotropes, all the microspores are held together to form a bag-like structure. So that condition is known as a pollinium. Okay, so here this is the way the tetrad of the pollens are formed. Okay, so that is a pollen grains. So pollen grains are formed from the microspore. The microspores are considered as a early development of the pollen grains. So we studied that microsporogenesis. Okay, so what is the pollinium? The pollinium is a very important 2-mark question. Okay, that is we can see in the calotropes. In the pollinium consists of 3 important parts. So one is the corpusculum, so another one is a retinaculum, and next that is a pollinia are present. Okay, so this type of arrangement is known as a translator. Okay, so this is the arranged as a inverted letter Y, so that is known as a translator. Okay, so where is the retinaculum? The retinaculum is a connect between the corpusculum and the pollinium. Okay, corpusculum and pollinium is connect with the help of the retinaculum. Okay, so that is a retinaculum. Corpusculum is a main branch. Okay, this type of arrangement we can see in the calotropes. Okay, so this is additional informations related to that. So next, cell wall formations in developing the pollen grains. Okay, how the cell wall is formed in the pollen grains? Actually, it consists of 2 types. One is a successive type, another one is a simultaneous type. So successive type means the after meiosis, actually the cell wall is formed. So after the meiosis 1 and 2, so in the monocot plants. Okay, so meiosis 1 is completed and meiosis 2 also will be completed. So then only that is a cell wall formation is occur, that is in the centrifugal manner. Okay, so this type of arrangement is known as a isobilateral tetrad. Okay, so isobilateral tetrad type is present in the monocot. Okay, and another one is known as a simultaneous type. So here the cell wall is formed after the meiosis 2. Okay, so that is after meiosis 2, the cell wall formation is occurred between the pollen grains, that is a tetrahedral tetrad. So that is the characters of dicot. Okay, so that is your one type is isobilateral tetrad, that is in the monocot, tetrahedral tetrad in the dicot plant. So there are other types other than this. Okay, this is a decussate, T-shaped. You can see that this is a T-shaped structure. Okay, this is a linear. Okay, all the haploid microspores are arranged in the linear type. Okay, this is a decussate. This is a T-shape. That's a linear. That is already we studied. That's the first one that is known as the tetrahedral, and second one is a isobilateral type. Okay, so this is a type of tetrad. Okay, so this is the way the cell wall is formed in the pollen grains. So next we are going to see the megasporogenesis. So megasporogenesis similarly, so the megaspore is also formed from the megaspore mother cell, so by the reduction divisions. So that is known as a megasporogenesis. Okay, you can see here that is a megaspores are formed. Okay, the single megaspores is formed, that is undergoing the reduction division to form a 4 haploid megaspores. From the megaspore the embryo sac is developed. Okay, so how the megaspores are formed inside the, that is a ovule. We know the structure of the ovule. Okay, so what are the portions in the ovule? It consists of the hilum, integuments, a micropylar region and chalosal regions are present. Okay, and inside that that is a sac-like structure is consists of a mass of parenchymatous tissues. Okay, the parenchymatous tissues are known as a nucellus. Okay, actually the megaspore is formed from the nucellus. Okay, in the nucellus also consists of your a hypodermal cells, that is we already studied in the microsporogenesis. A hypodermal cells is initiated that is a formations of microspores. Okay, so here also that is a nucellus. The nucellus consists of the hypodermal cells. Okay, the hypodermal cells act as a archisporial cell. Okay, so what is the difference between the microsporogenesis and megasporogenesis? Okay, so in the microsporogenesis, the archisporial cells are undergoing that's a periclinal division. Okay, to give primary parietal layer and primary sporogenous layer. Okay, similarly here the archisporial cells, it may be divided. Okay, it may not be divided. It may be divided, which gives rise to the primary parietal layer and primary sporogenous layer. But in some cases, the archisporial cells are directly act as here, that is a megaspore mother cell. Okay, so otherwise it may be undergoing the divisions. Okay, if it is undergoing the division, we can get it the two layers. One is the outer layer, one is the inner layer. Okay, but in microsporogenesis, the primary parietal layer which is undergoing the division, that is anticlinal and periclinal division, to form three anther walls are formed. Okay, but what about here? The three anther walls are not formed here. Okay, so what is the functions of primary parietal layer? Okay, the primary parietal layer which push the, that is the sporogenous tissues. Okay, sporogenous tissues are present here. The sporogenous tissues are pushed into that, say inside the nucellus. Okay, outer la irukkudiya and the sporogenous tissues ulla push panni nucellus kulla anappakudiya function yaarukku irukuna, the primary parietal layer ku irukku. Okay, so what about in the microsporogenesis? That is undergoing the division to form a three layer, but in the divisions agala which push the, that is a that is a sporogenous tissue inside the nucellus. Okay, because nucellus is supplying the nutrition to the, so the developing sporogenous tissues. Okay, so what about the functions of the primary sporogenous tissue? That is similar. Okay, that is similar to that, that's a microsporogenesis. Okay, the primary sporogenous tissues which is undergoing the mitotic division to form a, so that is a megaspore mother cell. Anga microspore mother cell form aachi, inga megaspore mother cell. Okay, the megaspore mother cell is undergoing the reduction divisions, that is a meiotic divisions to form a 4 haploid megaspores. Okay, the 4 haploid megaspores, actually any one is functional megaspore, remaining 3 are, that is a disappear. Okay, or any 2 is functional, remaining 2 is disappear. Okay, any one is functional, that is considered as a monosporic in condition. If any 2 is functional, that is considered as a bisporic in conditions. Okay, or trisporic or tetrasporic. Okay, based on the number of megaspore is involving in the formations of the embryo sac. Okay, so that is the difference between the microsporogenesis and megasporogenesis. So what I explained, that is everything is given in the short way. Okay, the hypodermal cells is a nucellus cells, archisporium, from the hypodermal the archisporium is formed. It may be directly act as a MMC, megaspore mother cell, or which is undergoing the periclinal division to form a primary parietal cells or primary sporogenous cells. Okay, the primary parietal cells is not going to form a 3 layers. So like in the microsporogenesis. Okay, anga mari 3 layer form agadu, adukku padila enna pannum appadina sporogenous tissues they have to embed the tissues to inside the, that's a nucellus. Okay, so primary sporogenous cells is undergoing the mitotic division to form a 4 haploid megaspores. In the 4 haploid megaspore la irundhu da namakku embryo sac start agudhu. Okay va, so how the embryo sac is developed அப்படினு பார்த்தோம்னா, so based on the number of megaspores, involvement of the megaspore. So monosporic அப்படினு சொல்லும்போது ஒரே ஒரு megaspore மட்டும் இன்வால்வ் ஆகும். If it is a 2 megaspores are involved, that is considered as a bisporic. If 3 megaspores are involved, that's a trisporic. All the megaspores are involving in the embryo sac development, that is known as a tetrasporic in conditions. Okay children, so what are the difference between that? Finally we can see some of the difference. The arrangement of the microspores and generations of the male gametophyte is termed as a microsporogenesis. Okay, it happens inside the microsporangia. Everything takes place inside the anther. That is, we studied under the heading development of anther. Okay, it has a many microspores mother cells that are distinguished into the microsporangium. So someone is that is separated from each other. In some species, that is held together. Okay, all the 4 cells of the microspore tetrads are useful. Everything is undergoing the tetrad formations. Okay, microspores வந்து tetrad எல்லாமே நம்ம யூஸ் பண்றோம். பட் இங்க பாத்தீங்கன்னா arrangement of the microspore and generations of the male gametophyte is termed as here. So that is a Microsporogenesis. Just you have to look at here, arrangement of the megaspores, female gametophyte from the megaspore mother cells, known as a Megasporogenesis. Okay, what we study, the same thing is only we are going to see here. They exist in the nucellus of the ovule, also known as a megasporogium, because nucellus in the nucellus the hypodermal cells are formed. Okay, so that is exist in the nucellus of the ovule is also considered as a megasporangium. So only a single megaspore mother cell is distinguished as a megasporangium. In some cases only one cell is involving to form an embryo sac. Okay, only one of the 4 cells, the megaspores tetrad, proceeds to develop. Okay, so this is just a difference between. I hope you could understand very well what are the difference between the microsporogenesis and megasporogenesis, and here you must keep it in your mind what type of division is occur in the part of the plant body. Okay, thank you so much.